Hello? Are you Sushmanth and Jyothi and Dick?
Hi, Bradley.
Hi, how are you?
Sorry, I'm fine. Sorry
It's
for the
okay.
disturbance. Right now, this week I haven't done any pretty work, Bradley. Actually, Mike told to stop the work and read one book which he have shared. I'm going through that book, like building a better project. It is mostly about building a better project using Pilint and other softwares. Yeah, I'm reading that book right now. Actually, a couple of days back, you have shared me some materials of C elegans. There are so many materials. I couldn't understand which order I should go to read. I mean, which order I should read about all these things. There is a book inside it, and different things are there. Can you suggest me an order of reading them all, which you have shared? Is it possible?
A what? Oh,
I mean, last week you have shared some materials about C elegans, right? For literature review for the paper.
yeah, we did. I did give you that folder, yeah.
Yeah. There is a problem. Like I couldn't understand from where to start. I mean,
Oh,
there are
well,
so many.
yeah, so I guess the way to read this is not to just read everything at once. Like a lot of the papers are kind of examples of things where people have used machine learning for C. elegans or specifically deep learning. So I wanted to give you some papers on things that people have kind of done in the past, but also in development. So they have a lot of the things like there will be a lot of terms like lineage trees and different places in the different cells in the embryo, things like that. And I don't want you to obsess over the terms right now. I just want you to maybe read how they did things and what they were doing, and then we can kind of step into the terminology more slowly. But it's something that you'll need to learn to know, kind of how like at least in C. elegans how a lot of the terminology for development, like for the lineage tree analyses that they do, and they do a lot. Sometimes they do gene expression analysis, which is even harder in terms of jargon. But I don't want you to get hung up on that. Just kind of forget about that for now, and kind of think about like what they did in those papers, like how did they segment cells, and how would they deal with a problem like a cell dividing into two cells. So
Okay.
that's where I kind of want you to go with that, and you can start at any paper, I think.
Okay.
But
I have read this paper called C Shaper.
okay,
It's interesting. It is mostly segmenting of cells and creating a 4D structure. I understood about it a little bit. Right now, there is a book also, and after seeing the whole folder, I was pretty confused from where to start. So I read C Shaper paper, but yeah, I will read them all for literature review, and I will implement my method, and then I will write what is different from my method and other methods. That would be fine, I think so. And yeah, I was like pretty stuck upon test cases, writing test case for the model. Mike told that he will hop into a one meeting and he will explain me how to do that. Uh, we, due to my own reasons and all, I couldn't attend the meeting. But he told like within 2 days we will have another meeting and we will resolve this.
okay. Well, that sounds
Yeah.
good. I have. I mean, that's something that I think it's coming along fine. Yeah, we had that meeting this last week where we
Okay.
talked about a lot of the progress that you're making, and so that's good.
And Jyothi I shared the folder with Jyothi Swaroop also. And then he's there in the meeting. That day is he only.
Okay.
He was reading those papers and he was trying to help me with the manuscript and the accuracy of the model, actually comparing different models. So yeah, my main motto is to write with Jyothi, me, you, Mayuk, and Mainak as team of writing this whole paper. This thing I have in mind, and we started work actually. We divided some papers between us, between both of us. He will read some papers and I will read some papers. Whatever I understood from my papers, I will explain it to him, and whatever he understands, he will explain it to me. In this way, we can easily defer work and do the work fastly. I was thinking like this. We'll try to complete as soon as it's possible all those papers, and yeah, I will start writing the abstract and all this stuff. Uh, you have any idea of publication and how to write? Please let me know about this thing.
Yeah. Well, I talked a little bit about how to write a paper, and
Yeah, actually, I was there. Jyothi Swaroop is not there.
Okay.
You don't know. If you don't mind, just could you explain it to me also?
Yeah. So, hello, Jyothi. How are you? He's there.
Yeah, he's there.
Okay.
I think he has mic issues.
Oh, okay.
Yeah, he has a mic issue. Sorry.
Oh, it's okay. So yeah, well, thanks for the update, Sushmanth, and yeah, let me go over the papers then, and we'll talk about some of the other things, like what we might do. So, Sushmanth and I met, I don't know, last week sometime. We talked about the sort of the project going forward, and then some of the things that he wants to put out, a couple of publications in a pipeline. So, like, he's got the idea of he wants to work on. So we have a paper, which is a kind of a preprint that's a very broad overview of DevoLearn, and then he wants to work on improving that, which is something we were trying to do last year, but we didn't get to it. And so that'll be something that we'll flesh out more. Then he wants to do a paper on like benchmarking, which is, I think, a good paper for if you were to submit to a conference or a journal. They always want to have benchmarks for your software to make sure that it's performs compared to something else. And then another paper, which I don't remember exactly what it was, but MedSam, yeah. So he's been presenting on MedSam, which is the SAM model, segment anything model, but like sort of for medical applications. But then we'd have like a version that would do things for developmental cells. So it's like take off on that and then writing that up. So that's good. To have that pipeline where you can identify the papers you want to write and then getting them off the ground. Basically writing an outline first and then moving on towards fleshing everything out. So I was talking to Sushmanth, this is for Jyothi's benefit, about how to write a paper. And the way we usually approach it is the classic introduction, methods, results, and discussion format. You have the introduction where you state the thing that you're doing, maybe the problems that you're going to talk about. Then you move on to the methods, which are all the methods that you use, and then the results, which may be like your benchmarking or like something like a screenshot where you're putting in like this is what our software looks like, and then discussion, which doesn't have to be long, but sometimes can be long, which discusses how you might use it. You might also have a use cases section where you talk about maybe one or two use cases where you're applying the software to different problems. So if you were to say, well, we could apply this to C. elegans embryo, but maybe also to other embryos. It's a different problem, so you have to kind of spell it out for people how to use these benchmarks or the software or whatever. The main idea here is we want to make, each paper would be for a different audience of readers. So like if you wanted to just know something about the software, maybe you weren't like a computer scientist even. The overview paper I think would be good because you can just kind of give people an idea of what it is and how to use it. And that's not going to be the same as like what computer scientists might want to see, which is a series of benchmarks, because they know how to use software. It's not, but they don't know why it's important to have it. have this software versus some other software. So that's why, the other thing about having these different papers is we want to write them up differently with the different sort of set of things going associated with them, and like maybe even a different kind of language. So like, cleaning up a lot of jargon that you might use in a computer science paper. So where we talk about, very machine learning specific terms, in the benchmarking paper, that's excellent. In the general paper, probably not, because people don't know what certain pieces of jargon are. And, you've read through those papers that I sent out on C. elegans, and you run into that a lot. You get like a lot of terms that, what does this mean? So that's always the problem we have with reading discipline-specific papers and things like that.
Actually, I have a small doubt. We have an end-to-end product, right? I mean, C Shaper, it was not hosted on different platform or something like that. We can use it just giving an image and extracting the features we want. But right now, the DevoLearn platform, right, which is there with us. Just a minute. Yes, I'm sharing my screen.
Okay.
Just a minute. I will rejoin the meeting.
Okay. Well, Susan, how are you?
No, I'm fine. I'm just at Clear Lake.
Okay.
You're on and off.
Great.
Very brief vacation.
Yeah. There we go. Hello, Lukas. Okay, this is render. This is, I think I've seen this before. This is for like live deployment, right? I think Sushmanth is having trouble with his audio.
Oh, sorry. Right now we have the end-to-end product which is deployed in render. Okay. Can't we write a paper on this? Like, we have built this software, we trained the model on ImageNet weights, and right now it is giving a good accuracy. There are three different type of models. One is the cell segmenter, membrane segmenter, and what they do. The whole paper will be about this. And actually, I have gone through all papers about CNGs. There is no this kind of software which will host on a platform and extract the features we want. So I was hoping, like, I have this thing in my mind, like, write about a paper about whatever software we built. Right now, DevoLearn is not working. I mean, there are some deprecated libraries. I will update it for sure, and make it working by the end of the publishing of paper. But can't we write a paper about this? Because see, whatever we have, three different type of models. There is a four different type of models actually. GANs is also there. It is just not hosted on the platform. I can do that. I can convert into a ONX type and I can host it on the Developmental platform here, and anyone can use. And I have gone through the whole. I have done some literature survey on the internet, but I couldn't find something like the software. I have this thing like if we run, we are getting. Sorry. Yeah. Shit. There is a small, I don't know what issue is. It is my internet. Yeah, I was thinking to write a paper about this. What we call explaining about the software what we did, and there is no software to compare. Also, there is C Shaper, but we need to run it in our local machine and show it like all these things. But this is a software which is hosted on the web. And I told to Mainak about this thing, and he was like, "Yeah, if there is no software like this which is hosted on a platform, why can't we write?" And this would be our first paper explaining and segmenting cells on web and showing it as an example, extracting a CSV file. I will add this feature also, extracting these points in a CSV file. Just download the CSV file and give it to a graph neural network and try to make the graph what we need.
All right.
I mean, it's a lot to take out, but yeah, I mean, we can prettily make this thing because I have seen on Hugging Face spaces, there is no, I mean, something like this which is hosted on a platform. So, my main motto would be like writing a paper on how we trained the model, on which weights we have trained, what methods we did, and how is the accuracy with the ground truths, and comparingly and hosting it on the software platform where someone can use it this thing and extracting the CSV file according to different time frames.
Yeah.
I hope we can do that, right? I mean, writing a paper about DevoLearn only, whatever we have right now, 3 models.
Oh yeah, that would be good. And like I said, that's something that we could decide whether we want to, at what level we want to write that. So we could actually write it at 2 levels. We could have something like that in the overview paper, kind of go into detail about it, but not really like, with a lot of the technical details. But that might be for like a paper where we benchmark things or like kind of really dig into the details of technical details of it. Because, we have people who you might want to use the software as like an online tool, and they might want to, they might have a familiarity with Hugging Face, but not with machine learning and programming. And then there are other people who want to actually contribute really deeply. So we want to have both of those open.
Yeah, mainly biologists because they don't know about the machine learning and deep learning things. They are mainly into biology and all this computation of cells. But yeah, if we provide something like this, they can use it right in a paper. That could I have this thing only in my mind right now. I was thinking about this, and I'm going through Minex code, what he did, etc.
Yeah.
Yeah, I was thinking to make the SAM model seem exactly like Minex and just create a comparison between these 2 models. I mean, which is hosted on a different platform, mainly technical details, how it is working, and all things. How zero shot performance will we go with the SAM, cell SAM, which I was thinking in my mind.
Yeah.
Okay.
Yeah. So yeah, that's good. Let me share my screen. Actually, I was going to go over some of the things. So as for other people's benefit too. So this is the paper I was talking about. So this is something that was written largely from a lot of the documentation and put together in a basic overview. So there's a short abstract, a summary, accessibility statement of need. So the statement of need is usually like, that's something that you can say, okay, this is why you would need this. And actually, the point you made about like this being hosted on the web, or being able to use it as something hosted on the web, is something we should add to this section. But it basically talks about why would you do this. Accessibility just means like where do you find it, which is probably should be up at the top. This is kind of a summary. So the abstract is kind of a very short summary of what happened. The summary is just what everything that's in the software. We'll update that with some of the features. Actually, we should probably break it down into the different toolboxes as well. We didn't really do that here, but that's because that's something that's been worked on this summer. That should be at the top in the summary. Then you have technical details. This needs to be filled out a lot more. Segmenting an embryo, and that's something that we can actually contribute more from, like other sources and other things we're thinking about. This was just a very basic description, and then maybe like a little demo, and then other things like hyperparameter optimization, lineage prediction, meta feature detection, and future directions, which obviously have to be changed. We've changed our sort of roadmap now, so we'll change that accordingly. But I mean, this is just kind of the structure of a very general paper where there is a little bit of jargon in here, but it's really kind of at a high level. And then a very deep level would be like a lot of the benchmarking, a lot of the, other things to consider, maybe some technical issues that are really kind of specialized, um, things that we've run into. I don't know if you've have a list of things that you have run into very specific to implementing some of these toolboxes. Like, we've talked about certain issues in the code and things like that. But it might be useful to kind of go through some of those issues. And I might also be talking about like maintenance issues too, because people come in, they want to know like what do we usually have to do to keep things running. So like obviously Python libraries always change and get deprecated, and it's just kind of hard to keep up with that. So, but
Yeah,
that's
right now
yeah,
I will work on this, Bradley. I mean, there is a direct integration AI tool which we can integrate with DevoLearn. It automatically um, changes the requirements.txt file according to what we need. That we can do it. And right now the update would be on the paper you wrote in ResearchGate was like this. We'll explain the more technical details about Resonate 18 structure, how we implement it. There are so many things that is. I have read that paper. Actually, it was there on ResearchGate, I think so.
yeah.
I read that paper. There are so many things not mentioned right now because we have update. We have hosted our things in Hugging Face also. Hugging Face, and we have the model right now which is working, and more technical details about what Resonate 18 structure, data pre-processing, because whatever data we have, there is something called noise in them. We added some different, we removed some noises, we done some image processing, and how we did the data pre-processing thing, and yeah, ImageNet weights what we used, and IOU score, etc. These things can be explained in a pretty clear way, and with that, we can mention about whatever work we did in the Hugging Face and how it can be used in like DevoLearn, this thing software. And our next future work in that paper would be like hosting it as a Hugging Face transformer, as a transformer library as a DevoNet. So yeah.
Yeah, that sounds great.
Yeah. I mean, in one paper, all three models will be explained: cell segmenter, cell membrane segmenter, and lineage population model.
Okay, yeah. So yeah, that sounds great. Yeah, whatever you come up with, I think is going to be pretty good. But we'll probably want to, spend some time thinking about how to structure this because, like I said, there are different levels of people will want to reach with this. So,
if you don't mind, I will share a doc with you, Mayuk, Mainak, and Jyothi Swaroop. And yeah, I will start writing out the paper. Um, every weekend you just review it and whatever changes need. If I write an abstract in that paper, just read that abstract, whatever changes I need to add, etc. You just mention it there or comment on there like you did for my GSoC proposal.
okay.
That would be fine, and I will start writing the paper after reading the documents which you have shared.
Okay.
It is okay. That thing is there. Yeah, that's my update. Pretty next work I will start training the SAM model and DevoNet model also. Yeah, I will create the comparison files, test files. I will have a meeting with Mayuk and I will give you update with me about all those things. And I will share the doc also.
Okay. Very good. Thank you. The other thing, I wanted to talk about with that is that I have a library of papers. This is stuff that we had like across the meetings that maybe in the last 3 years or so that we've talked about with respect to DevoLearn and some of the things that other people have done. So, they're like this paper, for example, is volume segregation programming in a nematode's early embryogenesis. This is from Physical Review E. This is some authors, and they've done this work on. This is kind of, I think, a more biologically oriented paper, but they've been doing these things with, segmenting cells and doing other types of fate specification and looking at model exploration. So this is maybe a bit too physicsy for what we're doing with DevoLearn, but it's important to read these kind of things to see if there are any methods that are useful, but also showing how people sort of analyze the embryo. And so some of these papers I included in here are kind of a little bit farther away from the machine learning applications, but it's definitely worth at least looking over. And we'll get into some of these papers later. Then there's this more accurate 3D atlas of C. elegans neurons. This is a paper where they actually take neurons, they segment the neurons out of C. elegans, set of images of C. elegans, volumetric images, and they're able to build an atlas. So the atlas building is, that's not really what we're doing, but a lot of the work that people have done with segmenting things, they're building things like atlases. So they're putting them into some other thing that they're, displaying on the web. So a lot of underlying, work that needs to go into, say, like an analysis at heart really involves like segmenting cells and doing things like that. So there's this other paper about establishing a morphological atlas of the embryo using deep learning based 4D segmentation. This one is probably the most relevant one to what we're doing, the C-Shaper. So this is what Sushmanth was talking about with C-Shaper. This actually is, the 4D is the 3 dimensions of space plus time. And they're working on this problem. And then there's a lineage resolved molecular atlas. So this is where you have an atlas of what they call molecular data, which is like the transcriptome. So it's like when genes are regulated by different processes, they produce transcripts. Those transcripts can be measured with RNA-seq, which we've mentioned in the meetings. And this is just basically a measurement of gene expression, and it'll vary by cell by cell, and you can actually build like an atlas where each cell has a complement of gene expression profiles, and you can then put this into things like UMAP or other dimensionality reduction techniques to get a map of what those should look like, and also put them onto the C. elegans itself. This is just kind of a paper about this other paper. So this is kind of an article summary. There's fast deep learning correspondence for neuron tracking and identification using synthetic training. So this is, kind of some of these methods that they're using, fast deep learning correspondence, or FDLC. They're doing this in C. elegans. I think this is like a technique they're working on. They're just using C. elegans because it's a simple system. But this is the kind of thing that, what we're doing with a lot of the deep learning here is we're trying to improve upon some of the previous techniques. And of course, the original technique was to hand draw the cells and the neurons and the C. elegans. So this was all hand drawn at one time. And then, there's different recognition approaches one can use. And so they're doing this fast deep learning correspondence, and it's basically a transformer, and they're training it on synthetic data, which means they're making data up based on the distributions you'd find in C. elegans, and then it predicts on actual samples of data. So this is something that, this is the way they're doing this. I don't know if this is useful, but it does give you some guidance in terms of what other people are doing. This is the same paper, I think. And then there's the one we talked about a couple points ago. And then this is a book, I think, but I think there's a chapter in here where we picked for C. elegans for 4D analysis. It's this paper here, chapter 2, automated acquisition of cell lineage through 4D microscopy and analysis of early C. elegans embryogenesis. So this is I think the paper that's relevant here. And this is, where they're doing this. Basically, they're constructing the cell lineage. They're using the data to do this. And this is a problem because we have the labels for the cells, but like attaching those labels to the cells and in images that are collected at different points in time, and then reconstructing all that is kind of a hard problem for someone who's just kind of starting out, or doesn't know a lot about C. elegans or whatever. So there's maybe some pointers in this article here, chapter 2 in this book. And then there's this zip file of spatial distributions for deep learning. So this is actually another, I don't know how many papers are in this, but that's another thing you might want to check out. So there are a bunch of papers in there that you'll find useful. So it looks like Lukas has a question. He has his hand raised.
Yeah, I was just wondering if you could share the link, like the Google Drive folder.
Yeah, I'll put it in the Slack for people, and then I'll send it directly to you, Lukas.
Thanks.
Yeah. So yeah, I recommend if people want to read it beyond Sushmanth and Jyothi, because there are a lot of good papers in there about some of these problems. So yeah, so Lukas is here and Sai's here. So Lukas has been working on, he's interested in contributing to some papers, and he's been working on some ideas. And so do you want to give an introduction, Lukas?
Yeah, so my name is Lukas. I'm an undergrad student in Canada, and I actually knew there's a lab called Orthogonal Lab, and it was virtual. Like I knew it from last year, but like I joined the lab this year, and I just joined the lab to look for opportunities, like research opportunities, and I'm also like I think I'll be able to present. Like it's like a presentation on biophysics, the biophysics of the bird flight, which I thought it's interesting. Not necessarily. I don't think it would be an interesting topic of research for because you need some facilities for doing. So, for example, what I was thinking as an extension to like the biophysics of the bird flight is like setting up an experiment to find like a in the lift force, like drag force, and these equations like the lift force, if you search it up, the equation is CL, which is a constant, times 1/2 rho v squared s. V squared is the velocity, s is the surface area of the wing, and this CL, which is technically like a constant number which varies from different angle of attacks and different different density, etc. I was wondering like this again is for the people who like orthogonology, but again you could set up an experiment to let's say find the CL, this certain constant number for like a certain bird species at like a let's say a different angle of attacks, and then publish it online, so the people like the ornithologists who are interested in this actual like stuff like the CL number could use it. And yeah, but it's not necessarily something that I found that we could do it, but again it's just a presentation that I'm gonna be doing tomorrow, and then like as I said, I'm generally interested in research opportunities. If someone has anything to present or like has any idea, I'd be interested to contribute if I see I could do it.
Yeah, that's great. Thank you, Lukas, for that update. So yeah, there are always opportunities to do things in the group here. We go over a lot of things during a meeting, and so don't feel overwhelmed. But also, if you want to reach out about something that is interesting to you, just please do, and we'll try to put something together. So a lot of the ideas we come up with are maybe things we can do, like as I think he's in the DevoWorm Slack. Something we can do is like an easy sort of research agenda. Sometimes it takes like a lot of effort to go and find the right resources and things, but we have to kind of get things off the ground. We have to discuss how to do them, and we have to give, just kind of the idea here is like present ideas and then work them out to some extent. And sometimes we get things completed, sometimes we don't, but it's always, don't hesitate to talk about what you're interested in. Yeah, I guess, yeah. So I think Lukas is in the Slack, DevoWorm Slack, and then so then please, get in contact. Sai, how are you? I've not seen you in the meetings before. Okay. Yeah, if you want to give an introduction, yeah.
I'm Harshitha, and I'm currently 4th year from Amrita.
Okay. Welcome, yeah. And yeah, the same thing holds true. Are you in our Slack, which is our communication channel for DevoWorm? If you're not, please put your email in the chat so I can, if you want.
Yeah, sure.
Okay. Yeah, that's great. So that's great. So again, the same thing holds true that I told Lukas. If you're interested in some topic and want to follow up, let us know. So yeah, I see that Dick put in the chat here that he dreamt all night about mapping differentiation trees of the genome. And then as an aside, anyone study graph theory, which is not networks, but I don't know if you wanted to talk about that more, Dick.
Yeah, sure.
Yeah.
Okay. Let me share screen here.
All right.
Okay, you see that one?
Yeah.
Okay, Bradley, this is for you to do. I found one tree, but it obviously doesn't show all 800 or 900 cells.
Right.
So we need one that does.
Yeah.
Okay, it's a pretty one. But
okay,
so that, and also I need you to check the cell counts in the paper.
Okay, the draft you sent me.
Yeah.
Okay.
Well, I just sent you another draft.
Oh, okay.
Okay, so here's what I got from last night. Okay, suppose we have n edges to a tree.
Okay.
Okay, then there are n factorial ways of ordering them along a linear genome. And this is on the assumption that each edge is retained intact on the genome. In other words, if they're split, it could be worse.
Right.
Okay. Now, are you familiar with synteny?
Yes.
Yes, okay. So I'm suggesting that synteny implies there might be some restrictions to this combinatorial problem.
Right.
But I'm not sure how you'd use it.
Yeah, so synteny is the order of genes on a chromosome.
Right.
Right.
Which seems conserved between species.
Yeah. And
okay.
Yeah.
Okay, so the mapping we're looking for should be reversible. In other words, you should be able to go from the genome and construct a tree from it, or from the tree to get its representation on the genome.
Yeah.
Okay, I mean, that's sort of obvious, but okay, now one thought I had is the metabolism of organisms has been worked out very well. Okay?
Yeah.
Now, metabolism involves enzymes, and the enzymes will have gene representations on the genome. Okay, so if you took a bunch of different metabolisms, you might be able to find a pattern in how the enzymes are distributed across the genome.
Okay.
Okay, so that's one possible location, one possible thing. And of course, I'm assuming here we're dealing with fully sequenced organisms such as C. elegans, where we know the whole sequence.
Right.
Okay.
Right.
Okay. Now, I started thinking in terms of graph theory. In graph theory, suppose you have a tree. You have a terminal node on the tree. Then each terminal node has a certain distance from the root, or from the start of the tree. Okay?
Okay.
So you could, for all of the terminal nodes, order them by distance. Okay? And that, if you take either the longest or the shortest first, that could place some restrictions on what the representation on the genome might be. Okay?
Okay.
So I think that's as far as I got. Yeah, that's.
What's the counting approach?
The counting approach is, I did look that up. The counting approach is to assume that the differentiation code is similar between different cell types, and you're looking for a pattern in the DNA that indicates the differentiation code. Okay, so given that, the number of copies of the DNA of that motif should be the same or approximately the same as the number of edges of the differentiation tree.
Okay.
And that's the counting problem. So if you can find a motif that corresponds to the number of edges of the differentiation tree, in other words, the number of cell types, then you may have the right thing. And I looked it up, and the number of cell types is far lower than the number of repeats of repetitive DNA.
Right.
Typically go into the hundreds of thousands. And, no organism is made of hundreds of thousands of different kinds of cells. So,
yeah.
So you might be able to distinguish it that way. Okay.
Yeah.
So I'm suggesting basically we might be able to find some sort of motif by first looking at metabolism, and then see if we can find a motif that corresponds in numbers to the number of cell types.
Yeah.
Okay, so this is obviously a massive bioinformatics problem.
Right.
Okay, but given the tree and given the genome, I think it's a soluble one.
Yeah.
That requires a supercomputer, but
yeah.
okay, because it's sort of hunt and see if you can find it.
Right.
Okay,
Well,
that's as far as I got. I think I just sent this to you and Susan.
Yeah.
Called it version 6.
Okay.
Okay.
All right. Yeah, that's
Any thoughts on this? I mean, you can see where graph theory is starting to come in.
Yeah. So, this is something, of course, you can do in C. elegans versus, say, like humans, because we know kind of what the cell types are in C. elegans. At least you think we know most of them.
I don't know if this is improved, but humans were estimated to have about 256 cell types. Well, this is 20 years ago.
Yeah.
And but mouse was estimated to have 5,000.
Yeah.
Okay. So it's, we don't have a proper differentiation tree of a mammal yet.
Right. Yeah, the whole thing about cell types in like mammals and these sort of regulative development contexts more generally is that you don't, like, you have a lot of precursor cells, you have a lot of things that are kind of type-like, so like, they're not quite, say, like,
and
not quite
also,
a neuron, but yeah.
if you dig into the literature, the definition of a differentiated cell is pretty vague.
Yeah.
Okay, so counting cell types when you can't say what it is,
Right.
is a bit difficult. Okay.
Yeah.
Some cell types are quite closely related to each other, and
Oh,
might
yeah.
be considered the same cell type, just switched in a different mode,
Right.
as in muscle.
Yeah, and you also may have what you might call cryptic cell types, where you can't distinguish them because you have no measurement,
Oh,
but they might be different.
yeah, that's fine.
That concept came out of the evolution of whole animals. They're cryptic species that don't interbreed, but they're I think this is in salamanders. They don't interbreed, but you can't tell them apart.
Yeah.
It's difficult.
Yeah. Okay. So I think C. elegans is the right organism because they have so much information on it.
Yeah, and it doesn't have as many cryptic cell types and things like that. Or we at least have a good idea of how to identify the cell types.
Yeah.
Okay. So yeah, I had some things actually to show about this. If I want to share my screen now, so.
Okay, I'll stop.
Yeah, so thank you for that. That's good. I saw a couple of versions come across the email, so I have I have those in my possession, so I'll take a look at them. I've been looking them over a little bit, but so we had a we had a question. Was that?
Just go ahead.
Oh, okay. So we had a question last week about like different types of like how cell types are regulated or switching between cell types, and so I put together some resources on this. So of course there's this literature on epigenetic switches and things like that. So I think this is where I put them. Okay, yeah. So there's all this literature on like these epigenetic switches. So I don't know if that's really what I want there. So there's these set of epigenetic switches that exist in the genome. Oftentimes they're associated with certain cell states, and so you have this. You have these CPG islands, these cytosine to guanine transitions, and there's a methyl group that sits on top of that, and it forms this epigenetic switch where it switches, what DNA is read during transcription, so that you have different. That's basically that controls different cell types. So when you have this state, the switch can be in different states, and it's associated. It's not they haven't necessarily demonstrated this, but it's always associated with different cell types. And critically, there's a bistability of the switch. So the switch can be bistable. It can switch from, like, say, a stem cell fate to a differentiated cell fate. And so this is one of the mechanisms that they talk about, these epigenetic changes. And so these are very important, at least in the literature, where they talk about some of these, when they look at, like, say, for example, cells that have been reprogrammed, where they look at the fate restriction of stem cells, they often assay for this. So this is a review article on epigenetic stem cells and epithelial cell fate, and it kind of goes through some of this, this work, where you have you can go through some of this what they call the histone code, which is a lot of the stuff that has to do with some of these epigenetic transformations. These epigenetic transformations play essential roles in the establishment of transcriptional programs accompanying cell differentiation. And so they talk about that quite a bit here. So that's just like, in different cell fates, say in I think this is mainly in mammals. I think in C. elegans they have, I don't know if they've established what the actual markers are, but this is another article, Epigenetic Regulation of Stem Cell Fate. This is on where they kind of go over some of the potential of looking at this. And they're talking about here the research that's been done so far is only the tip of the iceberg when it comes to writing an epigenetic instruction manual. So this is like something that people are kind of working towards, like saying, and this in this literature they have a tendency to think that it's all about this epigenetic aspect. So this is kind of one of these things where, if you don't include this in the paper, they get antsy. If you talk about cell fate. But anyways, sometimes people can fixate on certain mechanisms. But this is important for cell commitment and differentiation. So this is another paper. So I just brought up a couple of review papers on this. And then, of course, there's this epigenetic memory aspect. So there's this aspect of transitioning through cell types. So usually we have like a stem cell, and it's pluripotent, and it can maybe differentiate into several different cell types. And then so the fate is restricted to those cell types. And then you can differentiate it further into other cell types. So for example, you might go from like a stem cell to a neural precursor to a neuron of some type. And those are all transitions. And those are all kind of controlled by the transcriptome, but these markers will kind of determine that switch when that happens. That's the idea. So this kind of talks about epigenetic memory of the first cell fate decision, prevents complete embryonic cell reprogramming, embryonic stem cell reprogramming into trophoblast. So this talks about embryonic and trophoblast stem cells reflect the first irrevocable cell fate decision in development, and is reinforced by distinct epigenetic lineage barriers. So again, these epigenetic marks, they hypothesize are the things that are stopping it or can push it into a new fate. Nonetheless, embryonic stem cells can seemingly acquire TS-like or trophoblast stem cell-like characteristics upon manipulation of lineage determining transcription factors or activation of the extracellular signal regulated kinase 1/2 or ERK1/2 pathway. So there's this pathway that is involved in this. So they've interrogated the progression of reprogramming in ES cell models with regulatable OCT4 and CDX2 transgenes. Those are the genes that are involved in the stemness or maintaining the stem cell state. And so they're able to initiate differentiation, but lineage conversion remains incomplete in all models, underpinned by the failure to demethylate a small group of TS cell genes. So there's this whole model of how these things get regulated. You can do experiments to stop things from, differentiating and things like that. And then there's this paper on molecular hallmarks of epigenetic control, transition states and cell fate decisions in epigenetic landscapes. So this links this to Waddington's landscape, where we've seen Waddington's landscape where you have this landscape that's like a tree where it branches off. And those branches, of course, are the things we're trying to capture with different cell differentiation. And they kind of link what's going on in this literature to epigenetic landscapes. So if you go down to the, I don't know if they have an image of landscape here where you have a ball rolling down a hill into these channels, and the channels branch off so that you have these state changes, I guess. And so it's very metaphoric. This is supposed to be a gene expression network, but it's not really, Waddington's time, they didn't know a lot about that. So this is basically the idea. You have these two stable states on either side of this line, but on the line you have a metastable state. And the idea is that if it's in a steady state, it sort of can be seen in one of these stable states or in this metastable state where it's poised to differentiate. So it can attain one of two states by being in this metastable state. And there's this feedback loop here between X and Y, where these are the two cell states, X and Y, and there's this feedback that keeps it in the stable state, but there's also this break also that keeps it from converting to another cell type. When that break is removed, of course, you can go to the new cell type. When the break is applied, you stay in the same cell state. So there's this metastable state that involves both feedback and this break. So that's what they're talking about when they talk about a metastable state. It keeps it in this sort of, it keeps the potentiation there, but it's also, there are factors that keep it from transitioning to another cell type. Then there's this dynamic model, which is where you have this monostable state and then it bifurcates into this bistable state. So at some point, this stable cell X will hit this bistable switch, meaning that it can either remain X or go to Y. And so, or it's a stem cell that bifurcates into X or Y. So those are the two models they use here. And they're thinking about this in terms of Waddington's landscape. So this is, yeah.
So, yeah, these models look very much like the cell state splitter.
Yeah.
Okay.
Yeah.
So, we have metastability introduced by the intermediate filament ring.
Okay.
I have a question. So, you see on the bistable state, there's a continued dotted line.
Yeah.
If you look at, so if, let's say, this species, like, on the bistable, you see on the continued line, did it change the cell type or does it remain the same? Since it is a continued thing from the monostable state.
Yeah, so this dotted line is just kind of like, if you see up at the top in C, there's this metastable state. This would be the metastable state continued from like the monostable.
Oh, okay.
So like
so
the monostable
still doesn't change.
Yeah. So the monostable is like where you have a single cell, it's a stem cell, and it doesn't change to anything. And then you hit this switch point here where the red lines diverge. And it can either become one state or another. There's this regime in the middle, which is imaginary, but it's basically a thing that's keeping these on either side. So like, yeah. So yeah, they do mention like, they talk about the mechanisms for this in terms of like the cell state splitter analogy. They talk about things like chemical equilibrium and things like that, but no one knows really what the thing is that's keeping these things other than like an epigenetic marker, which of course is not necessarily, I don't think that's all that's happening, because it seems like there's, there's a lot more in the cell that's probably happening, but that's like one thing we can assay that's a quick and easy. So like this paper talks about epigenetic switching as a strategy for quick adaptation while attenuating biochemical noise. And so this talks about like some of the systems level things that are happening, and these epigenetic switches again are used as the sort of the stand-in for the stability or the ability to maintain like a bistable state or whatever. And then they talk about this in terms of environmental noise. So if you have a fluctuating environment or you have noise, in the presence of noise, this is something that reduces the amount of noise. So if you have this fluctuating environment, it can potentially push cells around to different states. If you attenuate that noise, you have a better chance of keeping it in that state. And the noises, of course, could be things like heat, or other types of things like osmotic pressure. Those are things that vary over time. So if there's a lot of variance, it can affect the cell and its state, its transcriptional program, and then it can push it into a state, maybe even into a cancer state. But what they're saying here is that epigenetic switching kind of mediates this, and they're using this as a marker. This is probably not the only thing that's happening, but there is actually a signature in the genome for where these epigenetic switches are. They call it a CPG transition, or a cytosine to guanine transition. And they have these CPG islands that you can that are in. I don't know if they have a CpG island map for C. elegans, but I know that they have them. They have characterized them in other genomes. And so this is, again, epigenetic and transcriptional regulations, prime cell fate before division during human pluripotent stem cell differentiation. This is, again, like priming cell fate. So people are always looking for how cell fate gets primed, or what the factors are that kind of started down that path. And they talk about this. This is like a method that they're using. People have developed all sorts of methods for like looking at these things. And the general thing is like, you'll maybe try to induce it with some factor, and then look at the transcriptional program before and after. And if you can find like signatures of that, not only the cell type but of differentiation. People haven't really looked at differentiation in that way, but like they'll look for different markers of a cell type, and they'll say, this cell expresses these genes, and they're upregulated, so that must mean that it's maybe like a muscle precursor, or it's a muscle cell because it's expressing these genes. And you can look at the morphology of the cell, or what the cell looks like under a microscope, but it's still kind of hard to, it's still kind of one of these things where you're just basically saying that it's a cell type because of these things, and it has these genes upregulated. So it's really kind of a, I don't think people have, and then this is probably not relevant, but I think that's like when you mentioned that last week, I had to go and look in the literature and see what the state of the art was in terms of what people are thinking about. So I don't know. I think like the idea about getting like motifs, that's probably where the opportunity lies. And then, even like having like a model of motifs or a model of some of these things in the literature, and incorporating into that model. So you can say, well, we have, we maybe can look towards maybe the way genes are organized on a chromosome, or different patterns of expression, different switches that might exist in the genome, and kind of making those kind of predictions for what might be controlling differentiation. Because there's cell type things that identify cell types, and things that identify differentiation. And it seems like the chromatin markers are sort of identify differentiation, but it's not clear like if that's in response to something else, or if it's just maintaining cell state. It's not really clear. So the literature is not set up to answer this problem, I don't think, but that's that's not a problem necessarily. I think people think about it differently in a lot of, like in the biomedical context, people think about it differently.
Well, ask ChatGPT.
Yeah.
I've asked ChatGPT a number of things, and I always ask for references. And that helps with a reference search. Sometimes it'll take you off into an area where, well, you need to go, but didn't realize it was there.
Yeah.
Anyway.
Any thoughts, Dick?
No, I don't have any thoughts.
Okay. Well,
No.
we'll keep working on it. I'll take a look at the draft and see.
Okay, I think, does anyone who's listening or in your crowd have bioinformatics under their belt?
Yeah, I don't think so, but but that's that's a skill we can. I mean, I know something about bioinformatics, I guess.
Yeah, I do too, but I'm not experienced.
But I mean, that's something, I don't know, people are interested in learning about different techniques. It might be something to go over in real time, maybe. I don't know.
Yeah, I mean, because for all the work we're doing, we're not taking advantage of the C. elegans genome.
Yeah, we should do that more. Yeah, it's not, genome, working with genomes is not necessarily easy. That's the thing.
I know that.
Yeah. Well, it's like raising, like sort of making conclusions from it, I guess, is the problem. Because it's like you have the sequence, but then what does that mean? We have annotations. They're not like, well, and C. elegans are more specific than, say, like in humans, but they're still,
Okay.
yeah. So, yeah, well, we'll revisit that. We'll keep working on that. Final thing I wanted to talk about was that this week we finished a newsletter for OpenWorm, so I wanted to go over that. So we're, of course, part of the OpenWorm Foundation, and this is the OpenWorm news for this month. So we sometimes put out, or it's actually Poreg Gleason, who's one of the senior contributors at OpenWorm. He's putting together these newsletters, and I've put in a part on DevoWorm and everything. So, so we have these, and this is so for June 2023. Looks like there was a poster presented at the C. elegans conference. So the C. elegans conference is a conference on people who are interested in using C. elegans as a model organism for research, for looking at things like disease, and for looking at connectomics and basic research. They get together at the C. elegans meeting every 2 years. And so it's really good because it's like all focused on C. elegans. Just about any topic you can think of. And so that it's hosted in Glasgow, Scotland every 2 years. It used to be in Los Angeles, but they moved it. And so they basically, this is the poster. We have, I don't know if this is going to open. I think it will. So this is the poster. We have the introduction where we talk about OpenWorm is an open science initiative where we have open data code and people can run OpenWorm as a simulation. They can, use the data, things like that. So we get the data from different sources. We've made data available from different labs, and we've talked about having an internship where people from different biological labs learn how to build open data sets. But those are things that, all involving the OpenWorm Foundation. Then, there was this one, the first study that really kind of came out of OpenWorm was the accurate simulation of locomotion. So we have these models of locomotion. We know that, C. elegans has these sort of stereotypic movements that it does, and we can characterize them, link that to the electrophysiology, link that to the cellular circuits and the connectome and the muscles. So there are these different gates that we can see under a microscope. We can model them, and we have these things that are generating those movements. Then one of the things that we've been working on for a while is this Docker package. So Docker is a sort of a virtual machine for your computer. You can set it up on your computer, and you can set up this Docker container that we have where all of the simulations of OpenWorm are in there. And so you open it up, you run it, it'll run through the simulations one at a time. There's of course, the locomotion simulation. So it's like cybernetic, which is here. You have other types of simulations of the connectome, like like C302 and some other things. And then, you can run those in sequence and see kind of the worm in pieces, I guess, or in different subsystems. It's really nice. It's still being developed. And so there are a lot of other things going on. There's a Hodgkin-Huxley tutorial that happened. This is a Google Summer Code project that Poreg was a mentor on. And this person, Rahul Sankar, developed this tutorial for a Hodgkin-Huxley model, which is a model of ion channels in C. elegans. And C. elegans doesn't have the same set of ion channels we do, but they don't fire action potentials the way we do. But there's still a lot of things to learn with the Hodgkin-Huxley model for C. elegans connectomes and behavior. There's also NeuroPal, which is this tool that looks at like the gene expression in brains, in worm brains, I guess. There's the simulation stack. This is DevoWorm and DevoLearn. I just put together a graphic of the different areas of research we do. We have mathematical modeling, cell segmentation, and secondary data. And, this is kind of a graphic. It's a very cramped area to do this in, but, we just have our little feature here. We have these studentships where we get students to learn, to do something in the foundation. So it involves a small stipend. And if you're interested, the application process is at this link, openworm.org studentships. And it's just like, it doesn't pay very much, but you get to work on, something in the OpenWorm Foundation, try to improve it. It's kind of like Google Summer of Code. The stipend isn't as big, but it's still kind of a nice thing to put on your CV, gets you into a project. So we have worm body models. We have electrophysiological recordings for different cells. So in C. elegans, all the cells, including the connectome cells, have an identity. So those are all like identifiable. And then we also have single cell data for a lot of things. So this is electrophysiological recordings from the ASH neuron. So we have a specific neuron we can record from. That neuron, we know the connections to other neurons, we can record from those. We can put together circuits, which are connected neurons, and then we can map that to some movement behavior, say, which of course people have done in different papers where they look at, like, if a worm is moving forward and then backing up, what is happening in that little part of the connectome, that circuit. And so we can actually characterize this quite well. So this is stuff that we're working on with other labs outside of OpenWorm. And then, there's future work to be had. There's a lot of stuff. There's modeling things in NeuroML, which is a type of XML. So it's like a markdown or a markup language for like the connectome. Incorporation of sensory feedback between worm body environment and neural network. So this is where you're characterizing sensory feedbacks. The different types of imaging, like calcium imaging. There's a lot of new techniques out there for collecting data, and that's something we can sort of embrace as a community. So I think this is nice because it kind of gives you an update as to what's going on in the project. This just kind of talks about all of that in the newsletter. And then, we're always constantly trying to fund our activities. And then, the studentships were successful for this past year. So this last year's studentship was where there was this, we have this OpenWorm browser, which is a virtual browser of the worm. And so you can it's blender.openworm.org. And you can look at the worm from different perspectives. You can remove the muscles or the cuticle, and you can look at the nerves, and the neurons. You can put the muscles and cuticle on. You can look at the whole worm. You can zoom in. You can get inside the worm to some extent. This had to be improved because it's kind of dated now. So Tyson Wheelwright, who's I think a graduate student at the University of Wisconsin now, he worked on this, doing this in Blender. So this is nice. This model is open source, so if people are interested in working on it more, it's possible to create a fork and to work on it.
Sorry for the interruption. The studentship is only, could you apply as an undergrad student, or should you be graduate or PhD?
I'm not sure what the rules are on it. I don't see why not, but I think it's a larger way for students, though. It's like, because it really does teach you. You could apply if you wanted. I don't know what the conditions are, but definitely check it out.
Yeah, Lukas, I'm going to look into that too. I didn't actually know.
Yeah.
First time I'm actually seeing that, so I'm curious about it. We can all look into that. Yeah.
Well, I worked with Porygon last year as a co-mentor, and kind of, I don't think the rules are hard set in stone on that, but I'm not in the board meeting, so I don't know what they're thinking in terms of. But yeah, I would definitely follow up on that. Yeah, so then we have all sorts of different types of data that are available. We have this is another thing too. If people are interested in projects, there's a lot of data available for C. elegans. Now it's sometimes a little challenging to work with the data because it's in a highly specialized format, and you have to figure out what the variables are and how to use them. But it's definitely you could do a lot of projects with the open data that's out there in C. elegans. It's possible to do a lot of really interesting things. So that's another thing. And then the simulation stack, which is running this Docker container, and that's something that I think is underutilized by people. It's hard to run because you have to run it on the right machine. I've been trying to run a large language model on a local machine recently, and it's hard to get these things to run locally. So it's not it's not something that's easy to do, but it's something that I think if you have the opportunity to set this up and get it running, it's really cool. So that's all I wanted to talk about with respect to that. I just wanted to give an update on OpenWorm and what's going on there. Oh yeah, that's actually Jesse. He's in both of the Slacks.
Oh, my
Yeah.
bad. Okay. I know.
Yeah. Okay, so yeah, this is or I guess Jesse said, Lukas, let's look into that. Curious myself. I also have a bioinformatics friend who I may want to collaborate with or invite here. We mostly deal with genetics, I think. Yeah. Yeah, we do. Yeah, we do a lot of we've done in the past a lot of sort of informatics stuff. Genetic, working with genome data is kind of challenging because like I said, you have to have sometimes it's in specific formats. You have to get it out and map a lot of these things to your problem. And so just grabbing some gene sequences isn't really the only thing you need to do. It has to be, but that's that it's available though, and that's I think the key. Right.
Hey, yeah. Sorry, I was gonna say this is my other account because I have a whole bunch of devices and phones and I'm traveling all the time, so that's why I'm here. But yeah, I'm glad. I'm either about to or just posted the newsletter Slack in the Slack because I've been wanting to find something like that for a while, and I'm glad to see this one. I think the last update was September 22.
Yeah.
So I'm glad to see an update for this year, and yeah. That's cool stuff.
Yeah. All right. Anything else before we go today? All right. Well, have a good week, everyone.
Okay.
Yeah.
Bye.
Bye.
Yeah.
Take care. Okay, now I want to talk about a few papers on constructive connectomics. And so this is going to go over some sort of recent developments in connectomics with respect to C. elegans and some alternative neural systems. And as we'll see, this has implications for the greater enterprise of connectomics. We'll have 3 papers. I'll start with C. elegans. So the first one is this Frontiers in Systems Neuroscience paper. This is from 2021. And this paper is called Computing Temporal Sequences Associated with Dynamic Patterns on the C. elegans Connectome. All right, so let's go over the abstract. Understanding how the structural connectivity and spatial geometry of a network constrains the dynamics of the network. It is able to support an active and open area of research. We simulated the plausible dynamics resulting from the known C. elegans connectome. So we know the C. elegans connectome both in the number of cells, the identity of those cells, hence the function of those cells, and the connections between them. So this is an excellent opportunity to map neurons to behavior. And in a lot of ways, people do that already. But there are a lot of open questions, especially in developmental connectomes, which aren't quite like the adult connectome. For example, in the different larval stages, you have different regimes of connectivity, which differ greatly based on the environment of the worm. In the embryo, you have sort of the precursors of this connectome. So we have the cells and we have the connections, so we can at least make comparisons with a full complement of a connectome. So what they're going to do is they're going to use a recent model and theoretical analysis that computes the dynamics of neurobiological networks by focusing on the local interactions among connected neurons, gives rise to the global dynamics in an emergent way. So this is where you have these local circuits of maybe 2 to 3 neurons. They might control something like forward movement or backward movement of the worm. You expand those out to other subnetworks that interact with that, and then you expand that to the entire connectome. So when they say global dynamics in an emergent way, they mean you have these global dynamics within the connectome that go beyond any circuit, but that those emerge from the interactions between maybe different circuits or subnetworks. We studied the dynamics which resulted from stimulating a chemosensory neuron, ASEL, in a known feeding circuit, both in isolation and embedded in the full connectome. We show that contralateral motor neuron activations in ventral and dorsal classes of motor neurons emerged from the simulations. So contralateral is the opposite side of the organism, or across the midline of the organism. So you have usually like a right and a left orientation of the connectome, and contralateral is going from one side of that midline to another. So just as an aside, why don't we look at this spatial organization of the connectome? So in C. elegans, you have a connectome that actually has a couple of features here. So one is that you have this, I think we saw earlier in the meeting, we have the model of the worm. The connectome is generally up here in the head, but there's some neurons in the tail, and then there's a midline that goes down the middle, and there are some clusters of neurons along that. There are lateral line connections and things like that that are important. But this is what the connectome looks like in the adult. In the embryo, of course, the cells are clustered in one area and they kind of emerge in another area. They're different in the way they're organized. There's cell migration as the larval worm comes to be, and then the larval worm has additional ecological adaptations, or sort of facultative adaptations, as they call them, that respond to differences in environment. So the Dower stage, for example, which is between an alternate stage of development between L2 and L4, it has a series of adaptations to very dry, very poor environmental conditions, which result in a very thick cuticle and differences in the way some of the sensory neurons are wired because their inputs are different. So this is the way it's set up. And then, of course, because we have the head and the tail and the midline, we have this left to right symmetry. And this is the head of the worm. And we basically have the symmetry in the connectome between left and right. So every neuron that we have an identity for, like ASEL stands, it's like ASE and then L, and we also have an ASER. So we have two cell types, one is left, one is right. There's the symmetry throughout the connectome. Sometimes there's asymmetry depending on the neuron, but usually there's a left copy and a right copy, and those emerge in the embryo from terminal cell divisions that result in different neurons, and then those are put into place in the adult, and they're wired up. They migrate to their respective position in the adult, and then they're connected in this way where there's a symmetry for the most part, and that there's this output in the body of a left and a right hemisphere, and then they move accordingly. So, left neurons on the left side might control right hand movements. Neurons on the right side might control left hand movements. So that's the kind of thing we're talking about when they talk about contralateral and asymmetry and things like that. Okay, so one interpretation of these results is that there is an inherent, and we propose purposeful structural wiring to the C. elegans connectome that has evolved to serve specific behavioral functions. So this is exactly what I was talking about with respect to some of these circuits, that there's mapping from a small set of cells to a larger set of behaviors or a specific behavior. Now, this is not really controversial in C. elegans, but in larger organisms or organisms with maybe larger brains or different types of behaviors, more complex behaviors, this is not really something that we accept as easily. And of course, we don't see that in a lot of cases. C. elegans and some other organisms like annelids, which also have a very small connectome, you see these very simple behaviors emerge. We've also done things with Bradenburg vehicles, which are another example of like a model that's sort of C. elegans like, in the sense that it maps behaviors from a set of connections and units. So this is definitely, I don't know what we would classify this as. Maybe this is some sort of like class of sort of brain behavior model where you can actually discern the behaviors from the different from a very small number of neurons, where we can actually look at the neurons and their connections and define behaviors, which are pretty stereotyped, by the way. But I'm not sure if anyone's actually done that work to classify that. To study network signaling pathways responsible for the dynamics, we developed an analytic framework that constructs temporal sequences, or what they call T-seq, because everyone has to have a seq in their method. But this is temporal sequences, basically these time-ordered walks of signals on graphs. So what they're doing is they're building a graph, like our connectome. And that connectome has nodes and arcs. And so like any good network, we have nodes and we have arcs. Now what's interesting about this, and you know this from graph neural networks, is that there's this message passing function. So you have one node A and you have another node B and you have another node C, and a message may get passed from A to B and B to C and C to A, and so it completes this sort of circuit. But there's also this message passing aspect. So however the message is passed, it's very specific. So you have A going to B, B going to C, C going to A. That's a circuit where you have a signal that travels around this circuit, but with feedback when it goes from C to A. And so they're very specific orders that you can have these messages passed. And a lot of times this is used to reconstruct graph neural network type embedding. So this is some technique. But aside from this, what we're doing here is we're not just inferring a connection based on some, aggregate message that we're not defining, but we actually know that this is some sort of signal. And the signal then has properties over time, which you can actually do signal processing on and find different properties, spectral properties of the signal. And so this is actually what's happening in a neural network, because you have the signal. It's usually like, maybe a spike or a pulse, but sometimes, there's a whole sequence of like gradations to that signal. And so that's what you're actually doing with in a connectome. So we found that only 5% of T-seq are preserved between the isolated feeding network relative to its embedded counterpart. So this is where you have, in this case, they're talking about embedding in a different way. They're talking about taking the feeding network as a single sort of closed network versus a network that's connected to other things. So any connectome is going to be connected both to itself, to that ABC clique that I showed you here, and then to other things. So B is connected to this, C is connected to this, A is probably connected to other things I'm not showing. Those are going to be signals that come in and out as well. And so those are going to affect the sort of the behavior of that little clique. If you close it off, if you just consider A, B, and C, it's a certain behavior. If you add in signals to different nodes from other sources, that's going to affect it as well. So when they say embedded, they just mean this clique with respect to the rest of the connectome, the rest of the connections. And when they say isolated, they mean just that clique, ABC. And so this is, let's see. The remaining 95% of signaling pathways computed in the isolated network are not present in the embedded network. So this means that there's a huge difference between just considering a clique and just in considering the entire network. So there's this, what they're saying is basically that there's this emergent property that occurs from a small control network being embedded in a larger connectome. And I think that's, maybe it may seem a little hand-wavy or obvious, but it's actually important because we don't really think about that when we talk about circuits in the literature. We usually talk about a circuit and it's being responsible for X behavior. And the reason I brought up the whole thing about larger networks with more complex behaviors is because that's exactly what you have in larger brains. You have these, nominally you have circuits, but those circuits are connected to other things. And so in C. elegans, where you have 302 neurons, and I think a couple thousand connections at the most, you don't really, this problem isn't really very hard. I mean, you can have these cliques, these circuits that we can look at, we can isolate them anatomically, and they're relatively isolated in from a connectivity standpoint. But then when we get to larger brains, we have these things that are more connected, more fully connected, are hard to discern as like independent modules, and it's hard to discern their outputs because sometimes they have many, many outputs. And so this is the problem. You have these isolated circuits that we talk about in the literature all the time, and you have this whole brain or whole connectome approach. So, this is something they talk about here is the functional consequences of the dynome. So they talk about the dynome, and I guess the dynome is like the dynamic aspects of the connectome, which is really interesting because I've not heard that word used before, but they actually talk about it in the paper here, and it's kind of their point. So this is kind of going through some of these temporal sequences, going through the cliques, the sort of the geometric cliques, and then going through how these things can vary, and then going through their connections to the larger network, and then those temporal sequences increase in terms of their influence because you have more of them coming in to that little clique. So this is figure 1. This shows a reconstruction of the C. elegans connectome. So these are the edges in gray. They're the specific neurons here colored. And so they did some simulations here. And they show that the number of unique network spiking states increases actually over relatively few simulation time steps. So within 1,000 time steps, they're able to get a huge number of unique network spiking states. And I guess this blue function here, if we look at D, so the full connectome is the red curve, the lattice is the blue curve. So this is where you get like this difference between the small circuits and the whole connectome. But also, when they arrange it in a lattice, that means you're limiting the number of connections. So a lot of connectomes are organized according to sort of a small world, or at least a scale-free principle. And so if you've ever heard of this term, this is where you have no sort of characteristic number of nodes to something where you have sort of hubs and peripheral nodes. So this is sort of like a hierarchical network. A small world network is where you have hubs that are connected. So it's connecting other nodes in the network. And if you want to look at the distance between any two nodes in the network, that distance is reduced in a small world network because you have these connections between nodes or hubs, and then connections between peripheral nodes and hubs. So it's like traveling in a airline network where you have cities that are joined by hubs. And so getting from city to city is a very short set of hops, as opposed to something like a lattice where you have a deterministic number of connections for each node. Let's say 2 or 3, I guess. Well, this has 4, this has 3, this has 2. We can, but anyways, this is what on the right, this is what a lattice looks like. So a lattice is often used as sort of a control for these kind of networks. And the point here is that the number of hops you need to take to go from one side of the network to another is going to be dependent on its position in the lattice, rather than being able to find hops between hubs and then finding another node on the other side. So this is why you get this kind of result. And if you see the simulation, it extends out quite a bit. It finds a large number of unique network spiking states very quickly. This is, of course, where they're doing this. They're treating the connections as signals, so they're actually looking at the number of spikes here in the full network and then the randomized full network. And then they're also looking at these simulation time steps for these TSeqs. So these are the TSeqs. So this is a temporal sequence from ASEL to the VB and DB classes of neuron. These are where you have neuron number and simulation time steps. These colored lines are lines of the signals. So there's a lot of technical detail I'm not going to get into here, but you can see kind of how they treat these data. So I think that's enough for that paper. Now I want to talk about this paper, which is moving on to alternative neural systems. So this is actually getting to something we talked about with respect to cell fates before, and that is, what is a neuron? And so when we talk about C. elegans connectome, we talk about a simple system, relatively simple system, where we have neurons and we have axons and synapses like we do in, say, the human brain, but we have these sort of stereotypical behaviors. We have a much stronger link between neural circuits and behaviors and so forth. But then the question is, what is a neuron and what is a nervous system? And so this actually goes further to say that, you don't even need a connectome to produce some of these behaviors. So this is an interesting paper that focuses on ctenophores, sponges, and placozoans. And so in these organisms, they have something called a nerve net. They also have, they express the proteins for synapses, but they don't actually have the kind of synapses that you would find in C. elegans or in humans. So this is it's a different way to make a neural architecture. So how to make a neuron, a synapse, and a neural circuit? Is there only one design for a neural architecture with a universally shared genomic blueprint across species? The brief answer is no. 4 early divergent lineages from a common ancestor, from a nerveless common ancestor of all animals. So C. elegans and humans and, a lot of animals are in this part of the tree of life where you have nervous systems, where you have these specific nervous systems that we're familiar with. And then there are other parts of the tree of life in animals where you don't have this. You have different approaches to building a nervous system. So there are some really interesting types of, they call them neuroid type integrative systems. So there are a lot of different types of these in animals. And one way is, of course, the way we're familiar with. This other way is where you have, say, comb jellies with unique synapses. There's So one of these is a subset of neural nets and comb jellies with unique synapses. The second lineage is the well-known Cnidaria plus Bioteria. Of course, this is where we are. The two others are non-synaptic neuroid systems in sponges and placozoans. So this is non-synaptic. So you have some with unique synapses, some with just regular synapses that we're familiar with, and then some with non-synaptic systems that are also neural in nature. So by integrating scRNA-seq data and microscopy data, we revised the definition of neurons as synaptically coupled, polarized, and highly heterogeneous secretory cells at the top of behavioral hierarchies with learning capabilities. So that's a mouthful. That is, so let's see. If we have, so the first thing is, the first part of this definition is synaptically coupled, polarized, and highly heterogeneous secretory cells. So we talked about synaptically coupled. So, they're coupled in some way. Actually, I think synaptically coupled might be a little bit, because they did talk about how there's either these non-synaptic neuroid systems, but you do have to have connections between them. All right, so the network has to have connections. This idea of a highly heterogeneous secretory cell. So I talked about I think it was last week, about the secretome. And so what happens is that you have cells, and you see this with all neurons, but especially with neurons that don't have a conventional synapse. So first of all, you have like gap junctions that connect neurons. And these are electrical connections where the cells come into contact and there's this connection. So the cell can pass things in between the cells through this gap junction. Another way, of course, is through the typical synaptic connection or some sort of connection between them, a physical connection, which is usually a chemical connection. So you have a synapse here, you have, maybe it's an axon and synapse pairing, it could be something else. The third way, of course, then is secretions. And so this is where the cell secretes proteins out of its cell body and into the local environment. So these cells here, depending on their distance from it, will get a dosage of whatever is being secreted from the cell, and hopefully it'll respond in kind. So there are a lot of peptide systems or peptidergic signaling that works in a way like this, where it gets secreted or released into the environment and it gets uptaken by another cell. Sometimes this is restricted to something you find in a synapse. In C. elegans, we have something called juxtacrine signaling, which is where signals get sent out in protein form, and they get taken up by its neighboring cells, and there's this cell-cell signaling that happens. There's also paracrine signaling, which happens at longer distances. But this secretome is interesting because there's a lot of stuff that gets pumped out of the cell and into the local microenvironment of the cells or the nervous system. That's very interesting. And this is, of course, an alternate way of building a nervous system. It's not as efficient as the synapse, and it's not even as efficient as the gap junction. But there is this aspect of, where you have the signaling, and that's the important thing, is the signaling. So I want to label these. This is a gap junction, this is electrical. B is a synapse, it's chemical, and then C is the secretome, which is also chemical. It's less efficient than B or A. So that's kind of the point I wanted to make on that. So this physiological and then of course we have behavioral hierarchies, which means that you have behavioral control, you have control of some complex behavior, you have a set of neurons for one function versus another function. You can compose them with learning capabilities, which means that these can establish learning. They can respond in kind. You can have like Hebbian rules even in a secretome. You can have other types of connections where the connections are strengthened over time, and that's the basis for learning, at least. We think it is. This physiological, not phylogenetic definition, so they're not thinking about this in terms of the tree of life. They're just saying that this is the physiology, the things that these physiological systems have in common. separates true neurons from non-synaptically and gap junction coupled integrative systems. So this is where you have this definition separates us out from sort of true neurons and non-synaptic gap. We talked about this, executing more stereotypic behavior. So this definition actually can differentiate these. So this is where they're just kind of like saying no true neuron does this, or a true neuron does this. It's sort of like a no true Scotsman argument, but okay. Growing evidence supports the hypothesis of multiple origins of neurons and synapses. So these happen multiple places in the tree of life, or at least they originate there. Thus, many non-bilaterine and bilaterine neuronal classes, circuits, or systems are considered functional rather than genetic categories. So, like I talked about earlier, you have these genes that get expressed. You can get a gene expression profile. Those don't tell you necessarily what the function is. It just tells you what genes are upregulated. The functional definition is different. It could be related to the things that it's expressing, but those aren't sufficient to explain the function. Those classes of things that are upregulated or downregulated, for that matter. It's only a clue. It's only a marker of maybe like identity or function, but it doesn't tell you anything about like the causes of it or defines what's going on there. It's kind of a loose association, sometimes a correlation, but you have to keep that in mind because sometimes, when you identify something from transcriptomics, it can kind of conflict with some of the things we find from functional studies or from, they include microscopy here, but there's a lot that is conflicting between transcriptomic profiles and the sort of things we can see under a microscope in terms of function or in terms of structure. So, thus many non-bilaterine and bilaterine neuronal classes, circuits, or systems are considered functional rather than genetic categories composed of non-homologous cell types. In summary, little explored examples of convergent neuronal evolution and representatives of early branching metazoans provide conceptually novel microanatomical and physiological architecture of behavioral controls in animals with prospects of neuroengineering and synthetic biology. So this is where we can learn from these systems. We can maybe mimic them. We can build, something that we can modify them and see how they work, or we can build our own system with all of its features. Well, probably not all of its features, but this is a nice review. Kind of goes through this argument. They're making an argument here. They're kind of making exploring these different classes of neuronal type of different types of neuronal architecture. They cite George Streeter, who's wrote a book on neuroevolution. So there's this, aspect of neuroevolution here that they talk about. And so this is actually an example from pleurobrachia, where you have the subepithelial neural net. So you have these cells in the subepithelium. It's a network. And so it's not really like a centralized nervous system. It's sort of distributed throughout the body of the organism, and it's integrated into the epithelium. So it's sort of underneath the epithelial layer, but it's all throughout the epithelial layer. And so this helps with the function of this organism because this organism needs this sort of distributed processing. You have mesoglial neuronal-like cells throughout in and around the subepithelial layer, and then you have the furrow here at the bottom. And so these are receptors kind of coming out of the body. So you have these tentacles coming out, and they're bringing their signals directly into these nets. Whereas in other organisms, you have a system of bringing them in. You have relays and things like that. You have a centralized nervous system you need to bring things in. And so that's the difference. And there are reasons why they have this sort of distributed nervous system. I mean, we have, to some extent, a distributed peripheral nervous system. But that's a different system because it's connected to a centralized system. It's largely for sort of external peripheral processing and moving your arms and your legs and things like that. and having that kind of resolution of the body as a reference point. But this is actually where it's distributed throughout the body, so there are points in this network that maybe are local processing that is, can take advantage of the global processing of the entire network. So that's, an interesting paper. I'm not going to go into any more detail on that. I want to get to our final paper, which is this paper on constructive connectomics. And so how neuronal axons get from here to there using gene expression maps derived from their family trees. The abstract reads, during brain development, billions of axons must navigate over multiple spatial scales to reach specific neuronal targets. So this is where in brain development, axons have to find other neurons. So there's this idea of axon finding, and there's this axon cone, which allows it to detect signals and find other neurons to connect to. And so this is definitely where you have, where cells are, neurons are independent at this stage, and then they're trying to find their connections. If you've ever seen this in culture, it's a wonderful thing. You usually have these independent cells floating around the culture, and then you have these processes that extend out of the cell. So it looks kind of like this, where you have like cells that are maybe just becoming neurons, and then they have these processes that kind of come out. And again, they have these chemical signals that are secreted out from the cell. So it's sending out signals that decay at a certain rate as they get away from it. And these axons, these processes are sticking out, and they're kind of moving along these gradients of chemicals. So it's sensing some chemical in the environment. And when it hits one of these signal gradients, it follows it up to the cell, and it establishes a connection. And so this happens all throughout the nervous system. You get these axons that kind of find their target. And so, what's the key here is that the individual cells are secreting proteins, or secreting signals for other, like for axons to pick up. So they have this axon cone, which is sort of like the end of the axon and controls the growth rate and where it's going and its direction. So this is how axons find targets. And this is something that in evolution changes, because you can have target tissues in different places in the organism. Let's call this a target tissue. And you can have motor neuron axons, for example, that go down, and they have to innervate the target tissue. They have to find the target tissue. And if the target tissue isn't there, say, for example, there's a tail, and the motor neuron sends out an axon, it innervates the tail, and then that controls the tail with that motor neuron signal. It tells the tail how to move. Now that neuron will exist as long as the tail is there, as long as its target tissue is there. When you take away the target tissue, what will happen is that the axon will migrate out, but it won't find a target. So it either finds another target tissue to innervate, which means that you have plasticity here in evolution. So it's sort of an evo-devo thing where it finds a new target tissue. Or the cell dies, and simply you lose motor neurons for that target tissue and can't reuse it. So there are a lot of scenarios, and this is all spelled out in the Streeter book that I mentioned earlier. This is, where a brief, just to recap, axons are sent out. They find their targets. Either it's a cell or a tissue. If the target's not there, the axon withers, and then the cell may die if it's not connected to anything. And this is a lot of the processes of development that are involved in evolutionary changes in phenotype, in like morphology, or sometimes just, because it doesn't have a tail. And so, it's a developmental developmental defect and doesn't have a tail. So the motor neurons aren't there too. And you can actually look in a defined mutant to see this sort of thing. So this is all what they're talking about here, with axons and navigating to their targets. And so build the processing circuits that generate the intelligent behavior of animals. However, the limited information capacity of the zygotic genome puts a strong constraint on how and which axonal roots can be encoded. So this, the genome is actually playing the role of building these proteins from transcriptomes that actually allow for these signals to be sent out and then for this communication to happen. So it all goes back to what the genome is able to encode. If it can encode signals, if it can encode it at in time, or enough of it is produced, then you can't have the system doesn't work. So these axonal routes are controlled ultimately by the zygotic genome. We propose and validate a mechanism of development that can provide an efficient encoding of this global wiring task. So a lot of people have approached this wiring task as a matter of optimizing the wiring. So the one way you can do this to understand the wiring patterns is to optimize it with respect to the topology. So you find the shortest paths, where you find the least overlapping paths, and you assume that that's the way that this system will evolve because it's efficient. And so in some ways that's true, but in other ways, we have these other constraints that play a role in how these connectomes are constructed. The key principle, confirmed through simulation, is that basic constraints on mitoses of neural stem cells, that mitotic daughters of similar gene expression to their parent and do not stray that far from one another, induce a global hierarchic map of nested regions, each marked by the expression profile of its common progenitor population. So these are daughter cells that divide at the same time. So in other words, if we think back to our lineage tree, we know that, for example, if you have a mother cell, which is up here at the top, I'm sure I have that already. All right. That mother cell then produces two daughter cells or more. Sometimes there's proliferation here. Those mother cells will pass on gene expression to its daughter cell. Sometimes it passes on the RNA in the compartment. Sometimes it passes on the ability to transcribe things. And of course, if you have a precursor cell here, then this precursor will constrain the daughter cells here, which are maybe more mature cells. The constraint is set here at the top, and it constrains what the daughters are doing. But it also sometimes mRNA gets carried over compartmentally in the cells, so that this is important, like in labeling the cells, but it's also an important point in what gets upregulated, what gets downregulated, things like that. So there are definitely constraints based on cell lineage. And so that's what they're talking about here. And this induces a global hierarchical map of nested regions, each marked by the expression profile of its common progenitor population. Thus, a traversal of lineal hierarchy generates a systematic sequence of expression profiles that traces a staged route, which growth cones can follow to their remote targets. So there's the staged route of cells and their descendants, and this usually happens in space. There are different ways that this happens, like there are different patterns of birth of cells, and they migrate in a certain pattern, and the growth cones of the axons can follow those growth patterns because they're actually set up chemically in that way. They're consistent chemically, and then there's a pattern there for the growth cone to follow. We've analyzed gene expression data of developing in adult mouse brains, published by the Allen Institute of Brain Science. So the Allen Institute has a really nice set of mouse brain atlases, which show not only the anatomy in high detail, but the gene expression maps and their spatial distribution in high detail. So they've done this for, I think, the entire mouse brain, or a number of different mouse brains. So they've done this for development and for the adults. This is a good resource if you're interested in this more. And found them consistent with our simulations. Gene expression induced partitions in the brain into a global spatial hierarchy of nested contiguous regions that is stable from at least embryonic day 11.5 to postnatal day 56. So basically what happens is that you have the brain as it's developing, at least in mouse, you have these sort of hierarchical sections where, basically development is stable. It allows for development to proceed without just sort of this problem of search. So like, the axon doesn't need to search every possible combination. It just needs to find its way through the predefined path. And that makes it a lot easier for these processes to happen properly. And so, we talk about how development is a process that's very regular most of the time. And it's amazing how that happens. And some of how that happens is to have these constraints on what the developmental system actually needs to do to achieve a stable phenotype. We use this experimental data to demonstrate that our axonal guidance algorithm is able to robustly extend arbors over long distances to specific targets. So your axon is like a tree, it arborizes and, that sort of thing. So it's doing this as well as like finding targets, and that these connections result in a qualitatively plausible connectome. So they're actually modeling this and showing that it does produce the kind of result that we would see in a real live biological connectome. So they're doing simulations here. We conclude that paradoxically, cell division may be the key to uniting neurons in the brain. So this is the paradox: if cells divide, they actually keep the brain together. So this is an interesting paper. I hadn't thought about it in this way before, but this is something they call constructive connectomics. There are two aspects of the construction problem: the generation from a few precursors of the vast number of various types of neuron that comprise the brain, and the process whereby these neurons connect to one another. So it's generating neurons and connecting them. The process of generation is well understood. We have lineage maps, fate maps, and lineage trees that we can use. But this process of connection is the harder problem. So this is what they're talking about. And so they're using these. They have their lineage tree here of cells, proliferation, axon navigation, and then you have this connectome once everything's connected up. So that's the problem. And that's their solution, is this constructive connectomics. So I think those were a nice set of papers. Thank you for watching, and I hope you learned something.